already virtual, and the winter time tends to be a little bit of an off season for MES anyway, so this might be something fun to try. The webinars have really been a hit so far. Every single one has been a lot of fun. People that have come have asked some wonderful questions. And as I said, I'm just so grateful that you guys all took the time to come tonight. So with that, I'm going to hand it off to Tom, who's going to give an introduction of Scott.
Good evening, everybody. Welcome to Scott Clem, all the way from Illinois. Nice of you to join us tonight. So Scott is a fifth-year final stage doctoral candidate, and he's a USDA NIFA predoctoral fellow in the lab of Alex Harmon-Threet over at the University of Illinois in Urbana-Champaign, also where I went to school and where I know Scott from. So he holds a bachelor's degree in zoology with a major in conservation and biodiversity, and he also holds a master's degree in entomology. Both of those degrees are from Auburn University. So while his dissertation is focused on the migratory ecology of Syrphids, he has a wide variety of other research interests that include insect ecology, conservation, pollination, biological control, IPM, taxonomy, collections curation, and toxicology. And for all of you who are hooked into the grapevine, he's currently looking for any job opportunities or postdoc opportunities. So if you know of anything, spread the good word. So tonight, Scott will be talking to us about some of the long distance migratory capacity of some of our North American syrphids. And with that, I will let you take it away.
All right. Thank you very much, Tom and Hillary. Yeah, this thank y'all for hosting me. I've been excited about this group. Seems like it's a good group. I've never been to Maine myself, but I'd love to go one day. Seems like a really beautiful area. All right. So yeah, like they mentioned, I will be presenting to you my a talk on a lot of the major components of my dissertation over the past 5 years. So I'm going to be talking about examining the long distance migratory capacity of North American hoverflies. Okay, so many insects are known to migrate in response to changing biotic and abiotic pressures. This can occur at local scales or broader continental scales, like with monarch butterflies, as you see here. I'm sure many of you are familiar. This type of migration often occurs in northward and southward circuits, in which the insects move south during the fall and north during the spring. But unlike classic vertebrate migrations like those of many birds, insects migrate using multiple generations. You know, insects are short-lived. They don't necessarily live for not as long as like birds and you know, things like fish and maybe mammals. So what you're seeing in this diagram, you see how insects will go through multiple generations in their round trip journey. And another thing that is kind of that the migratory insects tend to exhibit is what is known as partial migration. Now, this is a key concept that I'll be talking about throughout my talk. But partial migration is basically where the entire population does not migrate. So like some individuals migrate and some will stay local and overwinter. So this is pretty much exhibited among almost all known migratory insects. It is thought that this is a somewhat of a bet hedging strategy. So if you're a migratory individual, then you might have certain benefits if you move somewhere else, versus you know, and if you're a local individual, you might have other benefits. So insect migrations, they often take place at high altitudes where insects can take advantage of these swift unidirectional air currents that allows them to move great distances over a relatively short period of time. So recent evidence has been able to quantify these events and has demonstrated that we've basically just barely scratched the surface in terms of understanding the extent of insect migration. So this graphic here is from a paper by Galhu and colleagues, who used vertically pointing radar to show thousands of tons of insect biomass moving above, this was in the UK, annually. So if this would easily equate to the largest terrestrial migration on Earth. Yet to our knowledge, this behavior is currently very limited. Knowledge of this behavior is currently very limited. So monarchs and other Lepidoptera are perhaps the best known examples of migratory insect taxa, but there are several others. It is also known to be exhibited among several hemipteran species, such as aphids, plant hoppers, milkweed bugs, odinates like dragonflies, and coleoptera like ladybird beetles. But one group of migratory insects which have not received much attention, particularly in North America, are the Diptera. So what you're seeing here is a glimpse of the massive migratory movement of European hoverflies flying in a singular direction through a mountain pass in the Pyrenees. This is over in, like I said, in Europe. So these insects are likely migratory on many different continents, but they're by far most well documented in Europe. And this is partially because their mountains tend to be more latitudinally or more longitudinally based, while our mountains here in North America are more longitudinally based. So what you see here is how the mountains tend to funnel the movement of insects as they fly through. So you can really capture this massive migratory movement. But this is very concerning that we don't really know much about migration on other continents, given the importance of these insects. So hoverflies, as several of you know, are very diverse. There's about 6,000 species worldwide and about 800 to 900 in the United States. They are highly important in that they provide multiple ecosystem services throughout their life cycle. So they are pollinators, like almost all of the adults are pollinators of various crop and non-crop plant species. They're seen visiting and carrying around quite a bit of pollen. We often think of mostly bees when we think of pollinators, but flies are actually quite important. They tend to be underrated. They're important for pollination. They're also important in decomposition. So several species have larvae, like this rat-tailed maggot that you see here, that lives in dead and decaying plant matter, or like kind of lagoon-like situations where you have lots of dead and decaying things. So they're very important for breaking down nutrients and recycling them and whatnot. And a third ecosystem service provided by hoverflies is biological control. So what you're seeing here is a larva of the subfamily Syrphidae just munching away on an aphid. So these are actually quite voracious predators of aphids. They can knock out a group of aphids in a fairly short amount of time. So they're important to have around for that reason, also because aphids cause billions of dollars in crop losses every year. So this is they're quite important to have around. My goal of my dissertation has been to investigate migratory behaviors of North American Syrphidae. So what is the major significance of a migration with hoverflies? If North American hoverflies exhibited a long distance migration or migratory event, this would have major ecological and economic consequences. So in their regular breeding ground, they're of course important pollinators for a variety of plants. They feed on aphids, and they're also important prey resources for things like birds and spiders or things in higher trophic levels. So they provide nutrients like phosphorus and nitrogen and gigajoules of energy that are very important for the ecosystem. So if you are a migratory insect, you would be moving all of these ecological services with you. So you'd be moving it from one breeding ground to the next, where then you will be providing this to all the organisms in this second area. And also you would be distributing all of these ecological services in major stopover sites. So this is obviously, a very important knowledge to understand, so that we understand how these insects are important on a continental scale. So yeah. So as part of my dissertation, I have aimed it at understanding hoverfly migratory behavior. So I'm doing this using 3 overarching objectives. So objective 1 is to determine whether North American hoverflies migrate in the first place. My second objective is to explore potential behavioral, morphological, and physiological mechanisms of hoverfly migration. And then my third objective was to try and model natal origins of specimens and determine the extent of a potential migratory event. So for the remainder of this presentation, I'm going to be talking about these 3 hoverfly taxa, Alligator obliqua, Eupeodes americanus, and actually 2 species of Syrphidae. So all 3 of these are common throughout the eastern United States, anywhere between Illinois, and they're prevalent in Maine as well, and also in the southeastern United States. So these are all widespread. They're also voracious aphid feeders, so they're important for the biological control. So being part of that subfamily Syrphidae makes them important biological control agents. So we'll start with my first objective here. So this is kind of the opening act of my doctoral research, in which I just wanted to determine in the first place if North American hoverflies migrate at all. So you may be wondering, how the heck do you study insect migration? It's kind of difficult because they're short-lived. These are small insects, small organisms. You can't exactly put a transmitter on, especially the smaller ones like hoverflies. And even when you do, like there's some studies that look at dragonfly migration with transmitters. Even when you do, it could potentially impact their ability to move as far as they normally would. So there's obvious challenges to studying insect migration. But one way you can do it is to use stable isotopes. So stable isotopes are variations of chemical elements which exhibit unique atomic weights. Prevalence of the hydrogen isotopes protium and deuterium vary geographically in nature due to the differences in elevation, precipitation, and temperature. Biologists can use the ratios of these isotopes found in tissues, expressed as delta notation, to make inferences as to the geographic origins of these organisms. So for insects, these isotopic signatures become permanently fixed in wing and leg chitin after development. So as is illustrated on this map of dragonfly wing isotopes, an insect that developed in, say, Florida will exhibit an isotopic ratio value that is much less negative compared to one which developed in, say, Canada. One can then ascertain whether an individual is local or foreign based on its isotopic signature. So for this first objective, I sampled these 3 taxa that I showed you in Champaign County, Illinois, starting in the summer and going until the end of fall. So I sampled 10 specimens per species every 2 weeks. And a crucial part of this is to understand the local isotopic signature so that you can understand if individuals are not local. So in order to do this, I reared individuals in the lab on locally captured aphids to get an idea of this local signature. Yeah, so isotopes were measured by clipping off wings and legs of all these specimens. And I was able to derive their isotopic ratios using this comparative equilibrium approach, using the lab of my collaborator, Dr. Keith Hobson. So basically he was able to give me these, obtain these signatures and send them to me. Okay, so we'll jump right into the results here. So what you're seeing on this graph, this is the results of one species from 2017. What you see on this graph is that on the x-axis, we have our Julian week starting in July and going through October. On the x-axis, we have, the y-axis here, we have our isotopic ratio. So the more negative the isotopic ratio, the further north an individual presumably originated. And our black dotted lines here, this represents the general range of our known local signatures. So the range of what we would expect of that. So as you see with our first species, Allographta obliqua, it more or less kind of lies within this known local range. We get maybe one or two individuals here at the end of July that had a few negative isotopic signatures, but by and large, it seems to kind of lie within this range. The second species, Syrphidae, also more or less lied within this, this range, although crucially, during the expected migratory period of September, we did get at least maybe two specimens with some fairly highly negative values. But this pales in comparison to our third species, Eupeodes americanus, which exhibited very highly negative signatures at the end of August and through September. So, so this indicates that there's some sort of potential movement occurring in which we're getting individuals that may have flown into the area from a further northern latitude. There were even some values as highly negative as negative 181, which was very negative. So now we're going to move on to our second year's results. So in 2018, yet again, we kind of had Allographta that lied within this known local range, with maybe one specimen that was not very negative down here at the beginning of June, which is kind of an anomaly. We're not totally sure about that specimen. The second species, Syrphis rectus, this year, which was way more common, we found that it was highly negative. There were at least 5 specimens here during September and October that were highly negative. And then for the other species, Eupeodes, the trend wasn't as distinct as it was in 2017, but we did get some highly negative individuals here during September. So yet again, this kind of suggests that there were some individuals that could have been flying into the area during that time period. So what I think is happening here is that some individuals developed further north and flew south, where I intercepted them in Illinois. However, there's still much to understand about, why and how and where these insects are migrating. So why are they migrating? How are they able to do this? And also, where exactly are they coming from? So that's kind of what I am addressing with the rest of this presentation. So this leads me into objective 2. So here, I wanted to explore the potential behavioral, morphological, and physiological mechanisms of hoverfly migration. So I did this by answering 3 questions. I wanted to ask, does migratory hoverfly abundance change over time? My second question was, are migratory individuals morphologically distinct from non-migratory individuals? And finally, my third question here is, are migratory species more or less cold tolerant compared to presumed non-migratory species? So starting with question 1 here, I wanted to look at abundance. I noticed in 2017, when I was doing my first year's worth of data, that the Eupeodes americanus, the probable migratory species, was very abundant during the September time period. And this made me wonder, maybe I should try and quantify this. And so that's what I did in 2018, is I tried to quantify this abundance and try to see if we can piece apart if that has much to do with migration. So, so my hypothesis here is that abundance can be an indicator or a contributor to migration. Therefore, migratory species will be highly prevalent during certain time periods. So what I did is I conducted 25-minute hoverfly visual surveys in 5 different prairies in Urbana, Illinois. So these were the 5 prairies where I've been sampling hoverflies throughout all of my assessments. I recorded species abundance bi-weekly from August through October of these 3 species. And I also estimated flower and aphid abundance, because obviously abundance can be linked to multiple factors, such as resources like aphid abundance for the larvae, and then also, of course, flower resources that the adults feed on. So what I found here in this graph, let me kind of break this up for you to help you understand. The bars here represent the species. So we have Alligrapta obliqua in the red striped, Eupeodes americanus is in the green striped, and then surface, this is actually surface species, because you actually can't differentiate surface species in the field. You have to use a microscope. So these are all lumped together as one taxa here, but it's in blue here. So, and then also the yellow represents the percent flower cover and in these prairies, and then the purple represents the aphid abundance. These aphid abundance was quantified using sweep samples. So what you see is that indeed, Eupeodes americanus has a really high spike in abundance here right at the beginning of September. It may or may not be related to both aphid and flowering plant cover, because flower cover, this is actually mostly goldenrod, because goldenrod is a highly abundant flowering plant during fall here and across Eastern North America. It was starting to ramp up right there during the beginning of September. And aphid abundance had peaked right at the end of August. So this could be a potential response to aphid abundance. We also know that these aphids in this prairie may have not been the specific aphids that these hoverflies are feeding on, because there's also aphids in croplands and things like that that these could have been feeding on. So basically, aphid and flowering plant cover may have something to do with this abundance. But there could be other factors too. So it's interesting to think about whether or not these resources contributed to abundance, or if the migration attributed to this abundance. So basically, in this scenario, we would see that if we thought that the abundance was due to migration, we might expect that over time we would see these local individuals here, and we would expect an influx of individuals, which may cause this spike in abundance. However, it's also possible that this abundance caused a migratory event. So the idea here is that aphids and flowers are causing ideal situations for these insects. And so we have a very nice bloom in abundance. And because of this abundance, insects are having to compete with one another. So this competition tends to cause some of these individuals to disperse and seek new resources. So abundance, it's possible that abundance can cause migration as well. So, so it's interesting to think about it in that regard. Okay, so we'll move on to our second question here that's a part of Objective 2. So for this, I wanted to ask, are migratory individuals morphologically distinct from non-migratory individuals? My hypothesis here is that migratory individuals are subject to certain physiological constraints associated with long-distance flight. So therefore, they will exhibit morphologically distinct features. So this has precedent in the literature. When it comes to monarchs, for instance, it is known that migratory individuals have longer, more pointed wings than residents. When it comes to dragonflies, it's also known that migrants have a narrower wing apex. And even with Diptera, such as Drosophila or the fruit flies, we know that dispersers usually have greater body mass and larger wings that may decrease energetic and reproductive costs associated with long distance flight. Another key concept here is wing loading. So wing loading is another thing that can be related to migration. So this is a simple calculation that is the total dry body mass of an insect or whatever the flying object is, divided by the wing area. So the idea here is that the faster you go, the smaller your wings need to be to generate lift. So as an example, we'll use aeronautics. And this is a good thing that, this is what they use in aviation. So an X-15 supersonic jet can maximize its speed with smaller wings. And thus it has a higher wing loading. This also decreases its maneuverability though. But in contrast, like a Harrier jet might have a lower wing loading and more maneuverability. These same similar concepts apply to organisms. So if we pretend that these are insects, the X-15 here might be better at migrating long distances, but not as good at being maneuverable and doing things like escaping predators, like a individual that might remain local would be. The Harrier, in contrast, can maneuver and escape predators more easily, perhaps more easily, but may not be as well suited to a long distance migration. So yeah, so these concepts are known to apply to different biological organisms. So what I did to look at, understand whether morphology is related to migration in hoverflies is that I measured the wing length, wing area, dry body mass, and wing loading of the same hoverflies that I used in my stable isotope experiment that I discussed earlier. So by doing this, I was able to compare these morphological measures between species and also within species. So for the within species comparison, what I did is I took Eupeodes americanus, which is the most apparent migratory species, and I selected the individuals with the most highly negative isotopic signatures and designated them as migratory. And then I compared those to individuals with more local signatures as a non-migratory comparison. So with our across species comparison, what you see is that for wing area, starting with wing area, is that Allogratta obliqua has a smaller wing area and appears to be less variable than our other three species, which appear to be somewhat migratory, particularly Eupeodes americanus. So variability can be, by having a high variability, that suggests that there might be multiple phenotypes. So there might be like a migratory and non-migratory phenotype, versus if you have a less, a reduced variation, like with this species here, it might be that there's more of a singular phenotype. Yeah, so with wing area, we see that, like I said, Allogratta has a reduced area, and also it has a reduced wing length compared to the other species, and a reduced dry body mass, and then also a reduced wing loading. So, so even with this reduced wing loading, we see that the potentially migratory species have a higher wing loading, which would be what we might expect. When we do our within species comparison, we see that the migratory individuals of Eupeodes americanus has a slightly higher wing area compared to the non-migratory species or individuals. And this is statistically significant. It's also the case that the migratory individuals had a longer wing length, and a larger dry body mass, and also greater wing loading. So basically what this suggests is that those specimens that I picked out with the highly negative isotopic signatures don't appear to be just a random subset of the population, but a subset with morphological traits which may reduce costs associated with this long distance flight. So this is really the first potential evidence of migratory polymorphisms in hoverflies. There's a few studies in Europe that have tried to examine this, and have not found very significant results. So they didn't necessarily find this, but they also didn't use the exact same methods that I used. So just kind of a take home here is that our migratory specimens appear to have a larger body mass, a greater wing area, a greater wing length, and larger wing loading ratio. And our non-migratory specimens have a smaller body size, smaller wing area, shorter wing length, and a smaller wing loading ratio. Yeah, so this will lead us on into question 3. So for this, I wanted to ask, are migratory species more or less cold tolerant compared to presumed non-migratory species? The hypothesis here is that migratory species are less prepared for overwintering, so they will be less cold tolerant compared to our non-migratory species. So an important concept, so how do you measure cold tolerance in insects? One way you can do that is by examining the supercooling point. So basically this is the point at which an insect's body freezes. So if we were to, say, attach a thermocouple to an insect and look at its temperature and stick it in a freezer, we can measure its body as it decreases in temperature to the point where its body freezes. And there is a mild little bump here that's known as the heat of crystallization, which shows that this demonstrates that precise point at which the body freezes. And in our insects, it tells us the point at which the insect dies. So this gives us an idea of their cold tolerance. Also kind of makes us mad scientists by, you know, killing a bunch of flies, sticking them in the freezer and whatnot, but that's what we did. So for the methodologies, what we did is I went out and I sampled these hoverflies in the field. I brought them back into the lab and let them acclimate for about an hour, and then stuck a thermocouple to them in these microcentrifuge tubes, as you see here. I then put them in this polystyrene cube, which is calibrated to, when an insect is in the very center, as you see, it decreases at a rate of 0.5 degrees Celsius per minute when it's placed in a -80 freezer, like you see here. So I placed all these in this freezer, attached it to the thermocouple and the temperature data logger, and then I gathered the results. And I obtained the supercooling points of each of these specimens. So each line here represents an individual fly. And so this is what I found, is that, so one thing that may explain partially our supercooling point is body mass. So I incorporated body mass in the model of the statistical model here to help correct for that, or help include that as a potential factor. So even with that included as a fixed effect here, I still found that Eupeodes americanus, the migratory species, appears to have a decreased cold tolerance compared to our non-migratory species, Alligrapta obliqua. So this suggests that Alligrapta obliqua may be more prepared for a freezing temperatures and a scenario in which it gets very cold. Eupeodes americanus, however, might be allocating its resources towards a long distance flight. So this is interesting because it tells us this information. And this trend also holds in 2019. So I did this across 2 seasons and found similar results. Okay, so now we'll move on to my final objective here. So for this final part, I wanted to actually model the natal origins of these specimens using these isotopes and determine the extent of a potential migratory movement. So in 2019, instead of sampling in Illinois, I decided to go further south and obtain specimens from Alabama. So here I sampled Alligrapta obliqua, the presumed non-migratory species, and Eupeodes americanus, the presumed migratory species. And I collected these in North Alabama. To my amazement, several of these individuals of the suspected migratory species, Eupeodes americanus, exhibited highly negative non-local isotopic signatures during September, which is quite astounding. So I decided then that these specimens were perfect candidates for modeling their natal origins. So multiple steps are needed to do a natal origin assessment with hydrogen isotopes. Isotopic ratios change across trophic levels and ecological guilds and can be unique to specific species. Therefore, the first step is to estimate the rate of isotopic change, between water and hoverfly tissues and develop a calibration equation. We could then use this to convert water isotope values into hoverfly values and create an isoscape of estimated hoverfly isotopic origins. So a map of isotopes is known as an isoscape, like you're seeing here. So finally, using this special modeling software, we can predict natal origins of wild-caught individuals. So that's exactly what I did. So to establish this calibration equation, I reared hoverflies on 3 different types of water spiked with an increasing amount of the heavy hydrogen isotope deuterium. This drastically changes the isotopic signatures of those waters. And given that hoverfly larvae are aphidophagous, I use these waters to grow soybean plants and aphids, which were then fed to the hoverfly larvae. Upon pupation and eclosion, these flies were euthanized and their isotopes were assessed. We subsequently obtained isotopic signatures for the waters as well. So I then plotted the hoverfly isotopic values against their corresponding water values to obtain a calibration equation. This was done independently for both species because there are slight differences in the rate of isotopic change between them. So to create these isoscapes, spatial mixed models predicting isotopic values at locations throughout North America were initially created using rainfall isotope data gathered from the weather stations, which are these red triangles on this map as you see here. And this is overlaid on an elevation raster. I then applied this calibration equation and created a calibrated hoverfly isoscape. So again, this was done for both Alligator obliqua and Eupeodes americanus. So here are the outcomes of my natal origin assignments. So the way this works is that it compares the isotopic value of actual hoverflies with the predicted value on each cell in the isoscape. It then uses the Fisher's combination of probabilities method to assign origins to a group of specimens. On these maps, the red X marks the location at which the specimens were captured, and the green represents high probability of origin, while gray represents the low probability. So as you see, Alligator obliqua probably originated somewhere in central or southeastern United States, as you see here. Eupeodes americanus, however, appears to have originated much further north into the upper Midwest and into possibly up into interior Canada. So this is quite astounding as it indicates that some individuals could be traveling between 2,000 and 3,000 kilometers in a single season, which is quite amazing. So these results and those from my previous study indicate that like most other migratory insects, this is probably a partial migration. So oh, there it goes. Okay, so some individuals will migrate south upon the encroachment of winter, while others will remain local. And then overwintering offspring of fall migrants may then return in the spring via a multi-generational northward migration. But I haven't found any evidence of this yet. So, but it is known that they exhibit this among European Syrphidae. So, so it's quite possible, in fact, if not probable, that it also occurs here in North America. And so just to wrap things up, in conclusion, what my major findings are that some North American Syrphidae hoverflies exhibit highly negative non-local isotopic signatures during autumn and thus appear to be migratory. These insects are also highly abundant during the autumn. They exhibit unique morphological traits, and they are less cold tolerant compared to presumed non-migratory species. And then finally, natal origin assignments reveal that these insects are capable of moving at least hundreds of miles or thousands of kilometers from interior Canada to Alabama. So, so it really kind of sheds light that there is some sort of movement occurring here. So again, if we go back to our diagram that I showed at the beginning of this presentation, in knowing that these insects are migratory, they are likely distributing their ecological services far and wide across the continent. Farmers, gardeners, land managers, spiders, and birds too are likely, benefiting from this, and they expect these ecological services to be delivered to them during certain times of the year. And all of this is to say that understanding the migratory behavior of hoverflies is vital to conserving these insects and their ecosystem services. There's a few, future questions that I hope to answer going into wherever I'm going, if it be postdoc or job or whatever, if I'm staying in this area of research. But many things that still remain to be answered. There are many other species that are likely migratory, such as Aristolis tenax, as you see here. In fact, there's a paper published from the 1920s of this species moving along the East Coast, kind of where you all are, and over in Maine. This was actually in New Jersey, but presumably there would also be some indication of this. But there's been observances of this species moving in great number along that coast. So I'd actually be interested if any of you have seen any sort of indication of this movement, or maybe you can keep an eye out for it as well. Another question I'm interested in answering, are there regions in the US where these behaviors are visible? Again, I'd be curious if you all have seen this, or if you might be aware of certain locations. And then how do hoverfly populations differ genetically? So I want to take a population genomics approach to this and try to see if hoverflies across the continent really exhibit unique genetic signatures, or if they're all part of one massive genetic population. And then you can also, several other questions that can be answered using flight mills, which are basically where you put a little string on a fly or a bee or whatever, and you put it in this chamber, and you're able to estimate the length of time that it's flying, or the direction that it's flying, and you can answer a lot of questions about migratory phenomenon. So there are several questions that can be answered there. But anyway, with that, I'd like to thank my committee members, my lab mates, my isotopic collaborator, Dr. Keith Hobson, and then my hoverfly rearing support, Dr. Doris Lagos-Kutz. She's been very helpful throughout all this. And then finally, my funding sources here. I was able to get a SARE grant and also a USDA NIFA fellowship, which is what I'm currently on, to help fund this stuff. So, and with that, I'll take questions. I've got my email here and also my Twitter, so feel free to follow me on Twitter. And just like they said at the beginning, I'm looking for jobs and postdocs. So, never hurts to throw that out there. So thank you.
Great job, Scott. That was an awesome talk. We've got quite a few questions built up here in the chat. So I'm going to just go ahead and start from the top and work our way down.
Okay.
So the first question is from Kathy Clear, and she asks, did you take into consideration weather patterns? First objective, differences between the years.
Yeah, so weather patterns do play a huge role in migratory movements and such. There's been some situations where they're able to use radar to pick up movement of things like dragonflies following like cold fronts or things like that, or like massive wind fronts. So I sort of have tried to look at weather between years. And this has proven quite difficult because it seems like weather data is I don't know, there's just a lot of what's going on. And it's also the case that hoverflies it is known that they actually, based on European studies, that they can actually pick up on specific cues, like they fly during they're able to pick up on specific weather cues and take off. So if there's non-ideal conditions, then they won't be necessarily migrating. So I've kind of tried to look at this, but it's been kind of difficult. So so that's certainly something that that could be an indicator, an impactor.
Great. Andrea Couts, and I apologize to anyone here if I botcher pronouncing any last names or first names. Andrea Couts asks, are you able to make predictions about whether hoverfly migration is gradual with frequent stopovers or done in long stretches?
In long stretches. That's a good question. I am not totally sure how long an individual stretch might be. One method of potentially piecing that apart is through the flight mill, like I mentioned. But I'm not totally sure how many times they stop over, how many times they're long distances, they're like really long periods or short periods. I suspect yet again that has something to do with, you know, weather or potential like wind currents. Yeah, I mean, there are like specific southerly winds during fall that migratory insects are able to pick up on, which is why fall tends to be one generation while spring tends to be multiple generations.
And just a quick thing here, in case anyone here is not familiar with what a flight mail is, just because it's such a cool thing. If you could just describe that, that would be awesome.
Yeah, so a flight mail, it's basically a chamber where you can attach like a string to an insect, and it can still fly on that string, and you can observe whether or not it decides to stop or whether or not it decides to continually move. It involves magnetism. You're able to use magnetism to kind of, I think in some situations, it can allow like insects can actually pick up on magnetism and kind of know north from south and such. But yeah, so these flight mills are able to kind of give you an idea of how long insects will fly and also the directionality of their flight. So yeah, so certainly that would be an interesting way of piecing that question apart.
Awesome.
Yeah.
All right, Bob Nelson asks, is the next step, once you get your dissertation submitted, approved, and you're done with graduate student work, to collect this migratory species in central Canada to check for abnormally high deuterium, I can't say it.
Deuterium, yeah.
Ratios.
To see the highly negative values in Canada. Yeah, so there's kind of multiple ways of, so the way I did the isoscape was through an estimation method. So certainly you could go up into Canada and collect a few and kind of help verify my findings here. That would certainly be a good, interesting way of doing it, to see what those are like. Certainly over the summertime, or I guess in Canada, you would presume they're mostly local individuals. So certainly that could be an interesting endeavor in the future.
Awesome. And I think we'll just verify. I think that actually kind of answers another question. It's kind of cool. We got two similar questions here. From Mark, do you think if you were to go to interior Canada and measure the isotopic delta, that you would see a different range of lots of negative delta values?
Yes, basically, yeah. I would expect to see that from all that.
All right, we've got a less complicated questions. How do hoverflies overwinter? Great question from Jan.
Oh, that is a good question. So hoverflies overwinter as, well, it's at multiple life stages. We're actually, to my knowledge, we're not totally certain of these species that I'm examining precisely which stages are doing it, but based on European studies, it is known that hoverflies will migrate as larvae, pupae, and adults. So some adults will overwinter and emerge in the spring as a mated female, for instance. So like females, a fertilized female tends to overwinter and then will emerge with eggs and ready to lay them on aphids. So that's probably what our hoverflies are doing too, is something along those lines. But also there's kind of a bed hedging approach. So if a larva or if a pupa overwinters, then it might emerge a little later than the adult would. So it might be able to survive better, like a late occurring cold snap in the spring or something like that. So yeah, so probably it's the female adults overwintering, which is why I selected female adults for that cold tolerance experiment.
Great. All right. Last question here in the chat from Andrea. Since the migratory flies seem to actually have larger wings, do you think it matters more for a migratory fly to be able to fly longer rather than faster? Do larger wings allow for longer sustained flight?
Longer sustained flight. Perhaps. It's also that those energy reserves in the body that help an insect, fly for a longer sustained flight, I tend to think. And body and wing size tend to be correlated. So, yeah, perhaps, yeah, it's hard to say, if is speed more important or is it just that long flight? I would tend to think that evolutionary selection would select for specimens that are that fly a little faster potentially. So there might be like a kind of a race for who can get to the first location first, the new location first, and lay their eggs, and perhaps it's selecting for faster flighted individuals, but I don't know. It's hard to say, honestly. But that's kind of what my hypothesis is. Yeah.
We got one more question popped in the chat here. And anyone else, you don't have to ask the question in the chat. You're welcome to turn off your microphone and ask in person. So Kathy asks, do you think windmills might be an issue for insect migration?
Oh, windmills, like for sustainable energy stuff. I don't know. I tend to think that, because, obviously it seems to be a problem with birds and whatnot. So, I mean, it's definitely possible that windmills could be impacting insect migration as well. Yeah, it's really, I mean, yeah, it's probably some sort of contributing factor to some extent. But yeah, so I don't know. That's the answer to that question.
I see a few people unmuted themselves. Is anybody wanting to ask a question verbally?
If I might, I liked your presentation a lot, Scott. Your comment about Aristilus tenax was interesting because here in the Northeast, I'm in the Maritimes, and I'm sure this is a similar situation in coastal Maine. Aristilus tenax is super common in the fall, like October, November on asteraceous flowers.
Yeah.
They're there in abundance, and you see the same thing with Syrphidae militaris, which is interesting because that's not a super common species in general.
Yeah. Yeah, interesting.
So that's, yeah, like you wonder if those individuals are making their way northeast late in the season. And you wonder why as well. Like the species you talked about are aphid feeders, and it might make sense that they're moving around to look for aphid outbreaks.
Mm-hmm.
But if you're just looking for like cow patties or rotting whatever rat-tailed maggots enjoy, you think that would be less limiting? There wouldn't be such a great need for them to move around?
Yeah, it's definitely interesting. Yeah, why a decomposer might want to move like that. Yeah, I don't know. It could be just a competition thing, where it's just like a large abundance, and maybe it's just a. I mean, it's also a bet hedging thing. Aristilus tenax is a of course, it's a non-native species as well.
Right.
But
yeah. Does tenax, is it known to migrate in Europe, where such migrations might be easier to detect?
Yes, it is known to migrate in Europe. It's possibly the second most well-studied insect migrant, next to Episurphus balteatus, which we don't have here.
Yeah.
But yeah. So I was actually in contact with a guy in Nova Scotia who had he thought it was interesting that there were so many of Aristilus tenax there, that were just, they seemed to be there and then they'd be gone, so it'd be really interesting. This study from the 1920s, which is like it's only the There's only 2 prior studies on insect migration or on hoverfly migration in North America prior to my stuff, and one of them is that study from 1920 that looks at E10X in New Jersey. So yeah, that'd be really interesting next species to examine. Yeah.
Cool. It looks like Leslie's trying to get a question out over here.
Yeah.
Yeah.
Hi, I'm in New York City.
Hi.
And I'm just crazy about hoverflies. And I'm wondering, have you found they go to yellow flowers more than other colors?
Certainly they tend to like yellow flowers, and I tend to find that they visit a lot of white flowers. So like fall blooming frost aster is really popular. Really, they really like that. They like goldenrod as well. Kind of the #1 flower that I look for when I'm looking to sample hoverflies is chicory, which is the blue
purple
flower. Yeah.
Yeah. And bronze, they love fennel flowers.
Yeah. Oh, yeah. Yeah, I think I've seen them on fennel too. Yeah. Yeah. Chicory is. I don't know how prevalent it is around there, but around.
Oh, it is. It's everywhere. Yeah.
Okay. Yeah. Around here, it's like I look for a chicory bloom, and I go there, and I can find plenty of hoverflies, which is interesting because chicory is also a non-native species. But.
Okay. Thank you.
Yeah.
It's 8:04. Do you want to have a few more questions, Scott? Or are you?
Yeah, sure. I can answer a few more if people.
Got a couple in the chat. Okay. I'll pick one out of the 3 so we can end things up here. Let's see.
Yeah.
How about? Okay, that's interesting. Are stable isotopes available for testing from museum specimens of hoverflies, i.e., testing across decades and multitudes of sites? Is it a destructive technique for specimens? That's from John.
Yes, excellent question. So it is a destructive technique. So there have been lots of studies, or a few studies, that have used monarch and dragonfly museum specimens. Because those are bigger insects, it's not as destructive. You just have to get like a piece of the wing. Versus with hoverflies, you really have to lop off all of the wings and all of the legs. So it is quite destructive. But yeah, these isotopes, they're pretty much fixed in the tissues. So, so presumably you can get a specimen that's really old and and it still retains the isotopic signature of its natal origin. I don't know if the question has been answered that the older a specimen gets, I guess maybe let me rephrase it.